A genetic disorder is a genetic problem caused by one or more abnormalities in the genome, and that it could be inheritable if the parents have the genetic disorder in the germ line. The germ line would be like the cells that has the genetic information to be passed to the progeny. So if the parents have this mutation or abnormality, in the case of humans would be the sperm or the ovule, then the genetic disorder could pass to their sons. However, if you have like a new mutation or a new genetic disorder in a cell that is not the germ line, it won't be passed to the next generation. One of the most studied genetic disorders is one called aneuploidy. That is an abnormal number of chromosomes. For example, the Down syndrome is also known as trisomy 21. That has an extra chromosome number 21, as you can see in the image right here. There are many other kinds of aneuploidy. It could be one chromosome less or one chromosome extra. And these are very well studied disorders because we have been able to check these genetic disorders. I mean, we can do this experiment called the karyotype. So we can check how many chromosomes do you have from a long time ago. So it's a pretty well studied disorder. However, what is the difference between a mutation and a polymorphism? Let's see. A mutation is defined as any change in the DNA sequence away from normal. This implies there is a normal variation or allele. Alleles are different variations that could be different nucleotides variations that could be in a certain position in the DNA. This mutation, a mutation implies that there is a normal variation that is prevalent in the population, and that the mutation changes this to do a rare or abnormal variant. In contrast, a polymorphism is a DNA sequence variation that is common in the population. In this case, no single allele is regarded as the standard sequence. The threshold for this classification is 1% of the population. So if a certain population has more than 1% of a certain mutation, it's no longer called mutation, it's called polymorphism. And we have lots and lots of that in the human genome. One of the most common polymorphisms studied are those called single-nucleotide polymorphism. That is a kind of mutation or polymorphism that happens only in a single nucleotide. We have three kinds. We have substitution, that is the change of one base or nucleotide for another. We could have one insertion and one deletion. These single nucleotide polymorphisms, or SNPs or SNPs, could be within for example, a gene. What do you think could happen if we have one SNP within a gene? For example, let's see here. We have this the beginning of a gene. We have this is the translate one part that could be translatable. So if we have this first codon, this codon ATG, once it is in an RNA form, would be AUG, and if we see AUG, it's right here, it is translated into methionine. So this codon could be translated to methionine, this next codon could be translated to arginine, and so on. If this is the normal, with no SNPs, the normal gene, what could happen? What do you think could happen, for example, if we change this nucleotide here? Instead of being a cytosine, what would happen if we have a G, a proline? If we check the genetic code, the CCC codon and the CCG codon translates, let's look, CC, here we have CCC and CCG are in here, and both of them, both of those codons translates to the same amino acid, that is proline. We can see them in here too. C, 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 and G is for the same amino acid that is called proline or proline. So this kind of mutation is called synonym mutation, because the codon is translated to the same amino acid. However, what would happen if we change this first letter of the codon? Instead of being CCC, we have an ACC. So if we check on our genetic code, ACC stands for a CC here stands for threonine. So this, instead of being a proline, we have a threonine. This is a non-synonymous mutation within a gene that could modify the protein. Imagine you have a big Lego with lots of pieces and you change one. There could be relevant changes, or they could be not. It depends pretty much on the structure of the proline and the threonine and how much does it differs. And for example, if this part of the protein translates for a domain of the protein, remember that the domain is like an arm or some structure that does something for the protein, that have a function. It's like you have one less finger, imagine that. So there could be problems, there could be proteins with less efficiency on doing its job, or perhaps a better efficiency. That's how evolution works. What happens if instead of having a proline here, we have a threonine here in this protein? Something could change for good or for evil, so for worse. So everything else is okay. So this is what a substitution polymorphism is about. Could be synonym or not synonym. Next, what would happen if we have an insertion? As you can see, this string here is pretty much the same as the first one. However, there has been inserted one nucleotide here before the CCC. Before these 4 cytosines, we have added a guanine. So this kind of mutations are pretty much more dangerous. Why is that? Because as you can see, this changes this first codon. Instead of being CCC and being translated to a proline, this is translated to an alanine, and so on, because the codons, all of them are moved. So this codon here no longer translates for leucine but for proline, that is another amino acid, and so on until it ends. These kind of mutations and the one of the deletion, that is pretty much the same. If you delete one nucleotide, the codons, all of them are moved, so there could be mutations in all of the following amino acids. They could be wrong translations. So this is a problem because if you remember, we have several codons that tells the ribosome when to stop translating. As you can see, we have here these two codons, UAA and UAG, and we have these others, AGA and AGG, that that are the signals that tell the ribosome when to stop translating. So what happens if it finds a stop codon before it was supposed to? We could have a truncated protein. Or what happens if there's no stop codon? If the mutation is precisely in the codon where it was supposed to stop, there won't be a stop signal, so the translation would continue until probably the RNA ends or another stop codon finds. Or what would happen if we had a polymorphism in here in the initiation codon? So no initiation would happen in the translation process. So one single mutation could be very, very dangerous, for example in insertions or deletions. And substitution polymorphisms could be dangerous if it's non-synonymous. If it's synonymous, well, there's no big deal. What could happen? What do you think could happen if the polymorphism, or SNPs, or single nucleotide polymorphism, is not within a gene region but in a regulatory region? For example, in the promoter. As you know, there are several proteins called transcription factors that recognize a certain DNA string or DNA sequence, then interact with it, and this is like an activation of transcription of some gene. However, what happened if we have a polymorphism here? Does the transcription factor would find the string, or would it not? Or what happens if just it recognizes it less frequently? So transcription would be less frequent. It's not always like an on-off process. It's more like a continuous way of regulation. So if we have a mutation here and this transcription factor do not fully recognize this site, instead of having like 90% of affinity with this change, it could have like 70% of affinity to this sequence. It cannot well, the gene expression will be less. So will there be transcription, or would they not be transcription? Okay, well, this is all for our session number 2. Thank you very much. See ya. Stupid telephone.